So we've had a wonderful day of talks organized by our next speaker, Michael Hemann, and we're now going to hear from him. So he obtained his degrees from Wesleyan and PhD from Hopkins, and then spent several years at Cold Spring Harbor as a postdoc with Scott Lowe before he joined the faculty in the Koch 8 years ago, something like that, 2006. So Mike, great day, thank you very much. The floor's yours. You're going to talk about microenvironment and resistance.
Thank you. So I'd like to thank the speakers as well for the great talks today, and I appreciate having all of you stay to the bitter end. I will try to get through this quickly, if nothing else, but because the Zamboni arrives, I think, at 4:30, and you're all in trouble. He's a really unsettled person. So I'd like to talk about a distinct kind of tumor heterogeneity, and that's a heterogeneity that's actually introduced by the tumor microenvironment and not by mutation itself. Our lab is interested in how tumors acquire resistance to therapies, particularly frontline therapies, and our major focus has been on the study of hematopoietic cancers like Kim introduced earlier. These tend to be genomically simple tumors. There are also tumors that are highly responsive to frontline chemotherapy. So if you treat leukemias and lymphomas, they tend to go away very rapidly in response to frontline therapy. So after initial treatment, you are stuck with what's referred to as minimal residual disease, or at least a state where there's very little observable tumor burden. However, patients can also relapse with more drug-resistant tumors. And so really the effective or the significant challenge in treating these diseases is to eradicate this resistant or chemo-resistant residual disease. The question, though, is how is residual disease protected? What makes those residual cells distinct from the mass tumor burden, and how do we start resensitizing these cells to frontline therapy? Well, I'm going to present two mouse models that speak to the issue of not only residual disease, but therapeutic approaches that can be used to try to overcome resistance for that disease. And the first model is work done in the E-myc mouse, which is a nearly now 30-year-old mouse model of human Burkitt's lymphoma generated by Suzanne Corey and Jerry Adams that recapitulates the pathological lesion that's seen in human Burkitt. So these mice express MYC under the control of an immunoglobulin enhancer and develop B-cell lymphomas. These lymphomas, like their human counterparts, are really highly sensitive to frontline chemotherapy. But if you treat them with a single agent, the mice almost invariably relapse. So it provides us with an opportunity to look at the biology of response, residual disease, and then relapse. And so Luke Gilbert, a graduate student in the lab, a former graduate student in the lab, and looking at the biology of response and relapse, I think identified something very interesting about this model, and that is prior to treatment you have a very disseminated tumor, meaning the tumor's all over the place. But you don't see this at relapse. So if we treat with a frontline agent like doxorubicin, we get significant response, but a very focal response in this model. And the response in relapse is really localized to the mediastinal cavity, or the thymic, the mediastinal nodes that sit on top of the thymus. And you see a very different response if you look at these mediastinal nodes on the thymus versus the lymph nodes just pathologically. So if you look at the lymph nodes, all of these dark staining cells are just infiltrating normal lymphocytes. If you look at the thymus, you see a lot of these diffuse staining living tumor cells. So there's selective survival of tumor cells in the thymus following therapy. And it turns out the thymus is important for overall response and survival in these mice, because if we perform experiments transplanting tumor cells into athymic mice, RAG-deficient mice, which have an atrophied thymus, or thymectomized mice, we have an extension in overall survival. And so this really describes a heterogeneous behavior in a set of tumor cells that should be identical. And in fact, if we take tumor cells out of the thymus and transplant them back into mice, they go everywhere. So there's nothing special about those thymic tumor cells versus the peripheral tumor cells, except that they are selectively resistant to frontline chemotherapy. And again, in our case, define a state that we would refer to as minimal residual disease. Well, what makes these cells special? Well, it's not that they're less susceptible to DNA damage. If we just look at the amount of DNA damage they receive just as much damage in the thymus as they do in the lymph nodes. Here, just looking at gamma H2AX or phosphogamma H2AX staining, which is just a marker of DNA damage. And if we look by FACS analysis, there aren't a subset of cells with low gamma H2AX. So they're getting as much DNA damage, they're just not dying. And so to try to identify the basis for the survival advantage, Luke tried to reconstitute this effect in vitro. And so basically he treated mice, isolated all of their hematopoietic organs, diced them up into conditioned media, and examined the effect of various conditioned media from a variety of hematopoietic organs on survival of tumor cells exposed to doxorubicin. And what he found is that tumor cells exposed specifically to the thymic conditioned media, but not conditioned media from other organs, were protected from chemotherapy. So there's some soluble factors that are present in the thymus following chemotherapy that are inherently chemoprotective. And so to try to identify what those factors were, he performed cytokine arrays looking for factors that were upregulated and present specifically in the thymus, and identified a couple. One of them was IL-6, and the other is TIMP-1, which is an inhibitor of metalloproteinase that, as single recombinant proteins, could protect tumor cells from chemotherapy. Now, we next wanted to know what is the source of these cytokines. So just focusing on IL-6, he wanted to know, is it coming from the tumor cells themselves, or is it coming from the tumor microenvironment? And so to differentiate between these possibilities, he performed transplant experiments in which he transplanted IL-6 proficient tumor cells into either IL-6 proficient or deficient recipients and looked at the overall level of IL-6 following treatment. And what he found is he could only detect IL-6 in IL-6 proficient recipients following treatment, suggesting that essentially all of the IL-6 in this context is coming from the tumor microenvironment. And the survival benefit, perhaps, is coming from the tumor microenvironment. And so to try to see what would happen if you actually blocked IL-6 and TIMP-1 signaling, Luke looked at a pathway which is downstream of both of these factors, which is JAK-STAT signaling. Reason that if he blocked this survival signaling, he blocked the effect of this local paracrine survival signal. And in fact, that was the case. If he used a JAK inhibitor, he could significantly extend survival in the presence of doxorubicin relative to doxorubicin alone. Now, importantly, the JAK inhibitor had no effect by itself. It only potentiates the effect of doxorubicin. So you need something that essentially deprotects cells, and then you need something that kills them, because the deprotection in and of itself has very little effect. And in fact, when he added the doxorubicin and JAK inhibitor together, he could actually eradicate the thymic tumor burden, so essentially produce the same effect as not having a thymus at all. So he next wanted to know where in the thymus or what component cell type is responsible for IL-6 release. And so again, he performed a conditioned media experiment, but here he just disassociated tumors or component cells of the thymus following treatment to see where's the IL-6 coming from. And I think surprisingly, when he did this, he found that the vast majority of IL-6 is actually released from endothelial cells in the thymus, so vascular cells. I think we had assumed that it would be infiltrating immune cells, but in fact, it is component vascular endothelial cells. And in response to doxorubicin, you actually really increase this amount of IL-6 release from endothelial cells. And so following this work, he came up with the following model, and that is that chemotherapy induces DNA damage, and DNA damage eradicates the vast majority of tumor cells, except for in select microenvironments like the thymus, where not only does chemotherapy act on tumor cells, but acts on proximal cells like endothelial cells, and elicits, paradoxically, a pro-survival response. IL-6 and TIMP-1 in this context, that act on these proximal tumor cells and mediate their long-term survival. So you can overcome the effect of frontline chemotherapy by local protective signals that block or overcome the effect of that therapy in protected microenvironments. Now, importantly, the tumor cells in this microenvironment are getting just as much DNA damage as those peripherally. They're just not dying. And so we were able to show in conjunction with Graham Walker's lab that this in fact allows for mutagenesis of these tumor cells. So upon relapse of these tumors, you now have highly mutagenized tumors that are now intrinsically resistant to therapy. So initially you have heterogeneity mediated by a microenvironment, and subsequently you have heterogeneity mediated by mutagenesis that caused by selective survival in this microenvironment. Interestingly, this effect is not reserved for the thymus. We know now in work done by Eric Bent that we see secretory responses from endothelial cells in a variety of situations. One of the common links is that these tend to be endothelial cells that are in hypoxic microenvironments, including those found in tumors that seem to be activated to release cytokines in response to doxorubicin in a pathway that we know, based on work with Mike Yaffe's lab, involves activated P38 and MK2. But it's an effect that is much more common than simply isolated to thymic endothelial cells. Now, why do we have this kind of paracrine pro-survival effect? Well, I think we're still trying to figure that out. But at least one of our hypotheses is that this essentially serves as a survival signal to promote the regeneration of stem and progenitor cells. So following damage to an organ system, you want to have a cue that's actually going to activate progenitor cells to reconstitute that organ. This is actually seen in the thymus and has been a phenomenon that's established for a long time, that in response to whole body radiation, the thymus shrinks and then it regrows. And we see this effect is dependent in part on IL-6. So we think that DNA damage can actually induce acute pro-survival responses in select microenvironments, and tumor cells that find themselves in those microenvironments can essentially co-opt these signals. And so a therapy that targets residual disease not only needs to provide a frontline chemotherapeutic genotoxic signal, but needs to find a mechanism to essentially deprotect select microenvironments that co-opt these survival mechanisms. So the second model I want to present is work that was done along with Zhenzu Chen's lab at the KI and led by former postdoc Christian Palash in my lab, in which we were looking at essentially a humanized mouse model of B-cell lymphoma or leukemia. And Christian was interested in generating this kind of model because he wanted to use human-specific therapies like therapeutic antibodies. And so how do you humanize a mouse? Well, you start with human hematopoietic stem cells, and essentially you use them to reconstitute immune-deficient recipient mice. Now, if you want to create a tumor in this context, you have to put in oncogenes that are going to create a tumor. And in Christian's case, he started with lentiviruses that express a combination of MYC and BCL2, two really potent oncogenes in B-cell malignancy, along with GFP, under the control of a CD19 promoter. So you have B-cell specific production of these two oncogenes. And in fact, when you use these to reconstitute mice, you do get this kind of humanization. I don't know if this can be seen, but essentially you get human CD45 positive cells swimming around in the vascular system of recipient mice. Now, all of the recipients of hematopoietic stem cells with MYC and BCL2 develop tumors within about 100 days following reconstitution. They are looking at the red line on the Kaplan-Meier curve. If you get MYC alone or BCL2 alone or GFP alone, you don't get tumors. But the combination of these alterations produces tumors with complete penetrance. Based on surface marker analysis, we would characterize them as pre-B-cell acute lymphoblastic leukemias. These are a bit of a curious clinical entities because they are what are called double positive lymphomas or leukemias. They express MYC and BCL2. but that entity does exist in humans, and it's a particularly treatment refractory disease. Similarly, these tumors are quite refractory to therapy, so these mice fail if you give them frontline chemotherapies that are used in B-cell malignancies. And importantly, these are also transplantable, so they are bona fide tumors. They also look like the human disease, so we have these big blast cells in bone marrow, we have splenomegaly, we have infiltration in organs like the spleen with mitotic figures, and we also have infiltration into the brain, which occurs in human lymphomas and leukemias unless they're given prophylactic CNS treatments. Now, of course, the reason why Christian wanted to generate this model is he wanted to use human therapies, and one of the kinds of therapies that he wanted to use was an antibody therapy, and there are a number of choices. The one he chose was an antibody called alemtuzumab, which targets CD52. I'm not sure anybody knows what CD52 does, but it's on the surface of a lot of hematopoietic cells, including indolent B-cell malignancies. And it's an FDA-approved drug, and importantly, it's expressed really abundantly on these leukemia cells. And so as a single agent, he could get a significant extension in overall survival. Here looking at the red line as opposed to the black line, just as a single agent, although all of these mice relapse. This killing is mediated by macrophages, and we know that because if we treat mice with clodronate, which clears macrophages, we no longer get alemtuzumab-mediated clearing. Here looking at what's basically a log scale, so we get multiple logs of killing with just alemtuzumab, and we eliminate that with clodronate. It also requires both the Fc and the FAB portions of this antibody, because if we just add FAB fragments, we don't get killing either. And so presumably alemtuzumab binds leukemia cells, and then macrophages also bind the Fc portion and engulf these bound tumor cells. But of course, this therapy is failing. All the mice are relapsing in the presence of single compound therapy. So he wanted to know, what is the basis for failure in this context? And so he looked at a variety of organ systems following therapy. And so, for example, he looked at the blood, where again, looking at a log scale, we could get multiple logs of killing. So this antibody can clear tumor cells in the blood, it can clear tumor cells in the spleen and the liver, but it can't clear tumor cells in the bone marrow and the brain. So again, here we have a site of tumor persistence, a site of minimal residual disease. So we're having a heterogeneous response depending upon the site of tumor dissemination. So cells are selectively protected in this microenvironment. The question was, is the antibody actually getting to the bone marrow? Well, to answer this question, he looked at, or used a fluorescently labeled antibody and found that, sure enough, the antibody gets to the spleen. It doesn't get to the brain, as we might predict, so it's not crossing the blood-brain barrier, but it does get to the bone marrow. So you have antibody binding the tumor cell effectively in the bone marrow, it's just not clearing those tumor cells. So again, they're protected. So macrophages are unable to clear antibody-bound tumor cells in the bone marrow. And so how do we overcome this site-specific drug resistance? Now, one of the advantages of having this kind of model is we can do combination therapy, so we can look at a variety of agents that he thinks are going to promote clearance of these tumor cells. And so one of the agents he looked at first was GM-CSF to see if he could boost macrophage numbers to increase the ability of macrophages to target tumor cells, and this didn't work very well. And similarly, if you looked at other agents that you might use to eliminate tumor cells like radiation and other chemotherapies, he had very little effect, with the exception of the alkylating agent cyclophosphamide, which as a single agent was able to essentially eradicate, or in combination with alemtuzumab, was able to eradicate the bone marrow tumor burden. Again, we're looking at about 5 orders of magnitude. Similarly, it was able to essentially eradicate the tumor burden in recipient mice. So recipients of just cyclophosphamide and alemtuzumab alone as single agents showed some extension in tumor-free survival. Recipients of both were essentially cured. So these mice survived way out to over there. and were actually removed from the colony without disease. The mice that died initially died due to some toxicity mediated by alemtuzumab and cyclophosphamide, which is a concern with both of those drugs. Interestingly, though, we could dose reduce cyclophosphamide, so we could start at a very large dose of about 300 milligrams per kilogram, and then systemically reduce that dose to reduce toxicity down to about 100 milligrams per kilogram and still get the same effect. Now, this was curious to us because we think cyclophosphamide is a DNA damaging drug, an alkylating drug, and should function at high dose and needs to function at high dose to induce sufficient DNA damage to kill target tumor cells. But this low dose effect suggested that perhaps something else was happening. So again, we returned to a conditioned media experiment, isolated conditioned media from treated tumor cells in vivo, and found that tumor cells in response to cyclophosphamide actually release factors that activate macrophages. And we can see that in culture just looking at treated tumor cells where the tumor cells are green and the macrophages are unstained. And if you look in the middle, we get engulfment, effective engulfment of tumor cells by macrophages. There are two that come up from the bottom. It eats 4. And then it dies. That happens to dogs too, I think. But this is an effect that's mediated by essentially a paracrine secretory response from tumor cells. So what are the tumor cells secreting in response to macrophages? Well, they secrete things like CCL4, IL-8, VEGF, TNF-alpha. These are classic macrophage activators. And if we just look at the population of macrophages and tumor cells in bone marrow, we see we have lots of green tumor cells before cyclophosphamide treatment and some red macrophages. But upon cyclophosphamide treatment, we see essentially the coincident appearance of these macrophages engulfing tumor cells. This just doesn't happen this doesn't happen only in mice. If Christian looked at patients that are actually treated with cyclophosphamide, he sees a similar influx of activated macrophages or CD8 positive cells following chemotherapy. Now, importantly, the administration or co-administration timing of these drugs is really important. So basically, you have to give alemtuzumab coincident with giving cyclophosphamide, or at least one day from one another. Again, consistent with an acute secretory response from the target tumor cells mediating then subsequent alemtuzumab-mediated clearance. Now, what makes a bone marrow a protected microenvironment? We're still trying to figure this out, but at least one of the things that makes it protective is that tumor cells themselves are enzymatically responsible for synthesizing PGE2 from PGH2, and PGE2 is a real macrophage blocker. So it kills alemtuzumab-mediated killing, and one of the unexpected effects of cyclophosphamide is it really drops down the level of PGE2. So allows macrophages to be more active. And so the general model is that typically macrophage activity is blocked in the bone marrow until it's essentially deprotected by the presence of cyclophosphamide, which is having this really immunogenic role by not only eliminating the protective effect of the bone marrow, but eliciting the release of these cytokines that are activating macrophages that are essentially promoting the ability of macrophages to engulf target tumor cells. So I think both of these examples provide an example of how a site of tumor dissemination or invasion, and perhaps even metastasis in some cancers, establishes a heterogeneous response to conventional and even targeted therapies. And one of the real challenges, I think, in eradicating this disease is essentially deprotecting or eliminating these local protective effects of the microenvironment to homogenize tumors, to make them equally responsive to frontline therapies. I'll end there and thank all the people that did this work. I have a terrific lab full of fantastic people. We have excellent collaborators. I thank you all for staying here for this very long day. And I will turn things over to probably the best guy I know, Tyler Jacks, and I'm not, well, I'm sucking up, sorry. Yeah.
Thanks, Mike, for closing the show in such fine fashion. Thanks to everybody for staying to the end. We have a few minutes before the Zamboni gets here. I just wanted to make a couple of concluding remarks. Firstly, thanks to Mike for his organizational efforts in pulling this day together. He really did the lion's share of the work in that regard, and it was, as advertised, an outstanding day of science, really a terrific collection of talks, really important topic, and I think covered expertly by our group of speakers. So if you would please join me in thanking the speakers for today. You know, just I was thinking about what we heard about, and sometimes think about the history of cancer research going back, at least in recent era, to Nixon's war on cancer, and the sort of war on cancer theme is a little bit overdone. But I was thinking about sort of the warfare analogy as I was sitting there, and particularly the quote from Sun Tzu, the Chinese general who lived about 2,500 years ago. And you probably know the phrase, "know thy enemy." That comes from Sun Tzu. And the full quote is, if you know yourself and your enemy, you will win numerous battles. And he went on to say, if you know yourself but not your enemy, for every battle you'll win, you'll lose another. And finally, he said, if you know neither yourself nor your enemy, you're screwed. But knowing thy enemy, I think really that's what we're talking about here. And I think as the talks very beautifully described, the enemy in this case, at least in most cases, is quite complex. And it's complex for a number of different reasons, genetically, epigenetically, various program states, intracellular signaling states, interactions with the microenvironment. This is a complex problem. And yet we are making great progress, and that was also illustrated through the talks today. And I think the reason is that we're applying the knowledge of our understanding of the enemy. We're figuring out how it gets to the state that it gets to, the routes that it takes, the pathways that it uses to overcome our best efforts. And I think if you listen carefully, and I'm sure that you did, in every talk today, there was an undercurrent of, although this is challenging, we actually know how to get to the end and to get there successfully. So I'm uplifted, actually, by what I heard today. And again, I thank the speakers for their contributions. I thank Cindy Quentz and Pam DeFreya, Anna DeConnick, and Ann Chung for their efforts in putting this operation together. And finally, I thank you for being here once again. 14 years in, hopefully for some of you. 15th year will be probably back over in Kresge Auditorium. And we'll be talking next year about cancer prevention and early detection. See you next year.